November 17th, 2020 COVID-19 clinical rounds on critical care and especially focused today on convalescent plasma. My name is Richard Hunt. I'm an emergency physician who serves as senior medical advisor at the National Healthcare Preparedness Programs in the Office of the Assistant Secretary for Preparedness and Response at the United States Department of Health and Human Services. The purpose of these rounds are peer-to-peer, real-time sharing of COVID-19 challenges and successes. If I'm a clinician caring for COVID patients, the clinicians I want to learn from are those with the most experience. This is a voluntary public-private collaboration. And thank you to the multiple professional organizations, hospitals, and others who have collaborated on these rounds. They are not intended to be redundant or replace any organization's initiatives. They are intended to be sharing of clinical experience, not recommendations or guidance. These ECHO clinical rounds you are now participating in are focused on clinicians sharing their successes and challenges with other clinicians. Appreciation is extended to the National Emerging Special Pathogen Training and Education Center, NETEC, for their support with these rounds. NETEC has webinars that complement these Project ECHO COVID-19 clinical rounds. Thanks to Project ECHO, Dr. Bruce Struminger and his team for the peer-to-peer learning platform we use for these rounds. Upcoming ECHO COVID-19 clinical rounds currently are all US noon Eastern time. This Thursday, November 19th, in recognition of the surge and the challenges faced by those trying to work through alternate care sites, we're going to have emergency department rounds focused on the Javits Center, alternate care sites revisited. Stay tuned for announcements for next week's rounds. It is self-evident that our nation's medical response challenges continue to change every day. We'll continue to work to identify topics for these peer-to-peer learning rounds that are most timely and helpful to clinicians. Notably, Thanksgiving, we're going to take the day off, and you should as well, except for taking care of patients. So with that, I'm going to ask ECHO to go over some logistics, please.
Thanks. Hi, my name is Amanda Dzan. I'm with Project ECHO. Just a couple of quick things to go over before we really get started. Down at the bottom of your screen on Zoom, you'll see a Q&A function. Please use that for any questions or comments you have regarding the presentation or discussion today. There's also a chat function on Zoom. Please use that for any questions regarding technical or logistic issues, and we'll also be using that for the crowdsourcing question in just a minute. The session today is being recorded and your attendance is consent to be recorded, so we appreciate that. The presentation is not linked to the most recent email, but it will be available in upcoming ones down towards the bottom and on the ECHO website soon. We'll put a link for that in the chat in just a moment. Also, in about the last 15 minutes, we'll put a link to claim CME or continuing professional development credits. These are through the University of New Mexico, where we're located. The link is also in the most recent email announcement. And a quick reminder to everybody that the learning sessions are focused on improving patient care and health systems and supporting clinicians. So we appreciate everybody keeping it apolitical and non-commercial. A quick note on data use. We do collect data through the registration and polls and all the different ways that everybody participates, but just want to ensure everybody that we're keeping everything completely secure and only de-identified data is being shared with HHS and ASPR. The full little data security thing is right here on the screen. A quick note that NETEC has some webinars coming up that are complementary to the ones that we have here, and you can find those on the NETEC website. We'll also put that in the chat in just a moment, and there are two coming up this week. Also, a quick reminder to everybody that the AAMC has a continually updated COVID-19 clinical guidance repository that can be accessed on their website. We'll also share a link for that. And And back to the crowdsourcing question.
Thanks, everyone. Given the topic today, this is a pretty straightforward crowdsourcing question. Really straightforward. What has been your experience with convalescent plasma? What has been your experience with convalescent plasma? And even if it's like, haven't had any experience, knowing that, that helps people too. I have a few announcements, then I'm going to ask the panelists to take themselves off mute and introduce themselves, and then I'll introduce our speaker. So in terms of announcements, for everyone's benefit, want to announce that the Office of the Assistant Secretary for Preparedness and Response at HHS currently is procuring 30,000 high-flow nasal cannula kits for the Strategic National Stockpile that can allow its GM/Ventec V plus Pro ventilators to provide this type of therapy. This is the only ventilator model in the SNS with this potential capability. As part of the COVID-19 ventilator acquisition, the SNS now holds 30,000 Ventec V plus Pro ventilators that will be paired one-on-one with these new high-flow nasal cannula kits. For healthcare facilities in need of high-flow nasal equipment, they should work with their public health officials in their states, territories, tribal nations, or large metropolitan areas to request federal assistance through the normal processes for this. If the request is approved, the Secretary will direct the SNS to deploy the needed supplies to the Public Health Authority, which is then responsible for distributing them to the facilities in need. So again, high flow nasal cannula. And Amy and Amanda, if we have that link that we've used previously for the high flow nasal cannula, that'd be great to put in the chat so people have awareness of that. And then secondly, I mentioned next week, stay tuned. Lots of dynamics everywhere, including with the rounds in terms of like timing and so forth, so on. So just stay tuned as you see those emails to be aware of where we are with you. And then lastly, as I mentioned on yesterday's EMS rounds, we're actually at a once in a century disaster right now. And the rounds have that we start on March 24th. We're on rounds number 88 or 89. I literally have lost count. And given the over 36,000 participants that we've had on the rounds, it's timely to know if these have made any difference at all. Somebody's going to take a look at this in some history books and say, well, you did all that, we did all that together, 36,000 of us, and did it actually make any difference? We have a 4-question survey that we put up and used last week during the rounds. I'm not going to do that again today, but in your email somewhere, you should have received a survey. Again, it's 4 questions. There are a lot of surveys out there, but given where we are in history, it would be great if you could answer those 4 questions. 2 of them are quantitative, like we do in the quantitative polling on these rounds. The other 2 are qualitative, like, okay, it's a standard medical education type questions, like how did it impact your practice, those kinds of things. So at any rate, if you really could help us by responding to that, those 4 questions and those surveys, if you haven't already. Obviously, we're looking for double dipping on the results. So if you already responded to it, don't do a second time. So at any rate, really appreciate your participation in that if you haven't already. So with that, I'm gonna ask our panelists to please take yourselves off mute and introduce yourself, starting with Carol, then I'll go over to James Lawler. Carol, please.
Good morning. I'm Carol Cunningham. I am the State Medical Director at the Ohio Department of Public Safety, Division EMS.
Thanks, Carol, for being here. James, great to see you.
Hi, thanks, Rick. Good to see you. I'm James Lawler. I am an infectious disease physician at the University of Nebraska Medical Center, and I'm also one of the co-directors for our Global Center for Health Security.
Thanks, James. It's indeed an honor and privilege to introduce our speaker today, Dr. Nicole Bouvier, Associate Professor in the Department of Medicine in the Division of Infectious Diseases in the Department of Microbiology at the Icahn School of Medicine at Mount Sinai in New York. Nicole, over to you, and thank you for being here.
Thank you. I will just share my screen quickly. All right. Is it up? Okay. Great. Thanks for the invitation to speak today about the Mount Sinai Health System and our experience with convalescent plasma as a treatment for COVID-19. The Mount Sinai Health System is primarily located in New York City. We're a system of 7 hospitals, 6 of which are within the 5 boroughs of New York City. And this is our locations overlaid on a map of confirmed COVID cases in New York City neighborhoods. And as you can see, some of our facilities are located in fairly hard hit areas, including the main hospital, which is the large star here at the edge of East Harlem, Mount Sinai Queens here in Astoria, and Mount Sinai Brooklyn in Midwood. These were all areas that in the spring epidemic were hit rather hard. And so I just wanted to go over a little bit of New York's experience just to sort of set the context of our convalescent plasma treatments. This is the epidemic curve of new cases in New York State in blue and the city in red. And obviously the steepness of this curve is in part due to the inavailability of testing early on, and then also how rapidly New York City and state were able to ramp up testing capacity in the first couple of weeks of the local epidemic. But even so, the epidemic curve here was pretty intense. It's sort of like an epidemic cliff rather than a curve. And New York sort of ran face first into it. This is hospitalizations at Mount Sinai Hospital. As you can see, for the first half of April, we were over 2,000 patients census, COVID-19 only. This is in a hospital that is nominally an 1,100 and so bed hospital. So we were, over 150%, near 200% census for much of the month of April. And so, we faced a very unusual circumstance of, where do you put all of these sick patients? We had patients in PACUs and endoscopy suites. We turned some of our lobbies into patient bays. And we even had a field hospital set up in tents in Central Park. This is the view from my lab of Central Park, and the field hospital that was set up and run by Samaritan's Purse in conjunction with us. So it was an extremely unusual situation that we were operating in. And also, we didn't have much chance to get ready. In terms of the timeline of when our epidemic started, it was right around the 1st of January when word started coming out of China of an undiagnosed pneumonia illness, which subsequently was pretty quickly confirmed to be a coronavirus. On the 10th of January, the first genome was posted online. And thankfully, one of my colleagues here at Mount Sinai, Florian Kramer, who is a protein wizard and whose lab can pretty much make any protein anytime, took that genome and started working on synthesizing recombinant proteins, viral proteins for use in research and diagnostics. And so we had, fortunately, that head start as we were starting to see cases in the United States, first in West and then in New York City. First confirmed case on March 1st. We know now from some work done by colleagues at Mount Sinai through serology and also genomic phylogeny that probably there were multiple introductions into the New York City area in early February. They seem to have come primarily from Europe as opposed to Asia, which I think everybody at the time sort of had their eyes turned in the wrong direction. You know, we know that the virus was probably circulating under the radar for at least several weeks before it was actually diagnosed. And that was actually here at Mount Sinai, was the first officially diagnosed case. By March 17th, the Kramer Lab had published on medRxiv a detailed protocol for how to do their serology assay, which is a two-step assay. It has a qualitative step, which is just one serum dilution assayed against the receptor binding domain of the spike protein. And that's just a positive negative readout. Positives are then transferred into a quantitative assay, which is 5 serum dilutions ranging from 1 to 80 and 1 to 2880 against the full length spike protein. And at this time, the Kramer Lab was working with our clinical lab to transition this research-based assay into a clinical assay, which was sort of accomplished throughout the month of March and actually received emergency use authorization on April 15th. And so the transitioning of this antibody titering assay to the clinical lab allowed us to start screening potential donors for convalescent plasma. So the FDA announced on March 24th that they would be facilitating investigational use of convalescent plasma through several different pathways, IND, emergency IND, clinical trials. And we actually started screening for donors on March 26th. And that was headed by my colleague Anya Weinberg. We screened tens of thousands of New Yorkers ultimately throughout the whole epidemic. But really we started screening you know, only a few weeks after the first confirmed case. So early on, we were looking sort of for a needle in a haystack because there just weren't that many people who were convalescent at that time. But you know, we were able to start locating people who had infection and had seroconverted. And fortunately, the assay that the Kramer Lab developed allowed us to only refer donors that had a certain titer of specific anti-SARS-CoV-2 antibodies for plasma donation. And we have now seen through some retrospective work done by the Kramer Lab and others that the clinical assay, the serum dilutions of the clinical assay actually correlate quite well with the neutralizing capacity of the serum. And as the clinical assay titer gets higher, the neutralizing capacity increases as well. So we were able to sort of pre-select donors who had the highest levels of antibodies for donation. And then our first treatment happened on March 28th, was when we received our first shipment from New York Blood Center. And then again, to overlaying the epidemic curve here in the city and state. This is our histogram of convalescent plasma transfusions that we have done since March 28th, which was our first transfusion. We started under the EIND, or Emergency Investigational New Drug pathway for the FDA, where we submitted individual applications for every patient that we treated. They were approved by the FDA before treatment, and you know, we got informed consent from patients as well. That was our first 39 patients were transfused under EIND. We then switched over to the expanded access protocol, which was opened and led by Mayo Clinic, where it was you know, a more centralized application process where we enrolled patients in the EAP and were able to transfuse without requesting individual permission for each and every patient. We were at the same time planning to roll out to the entire Mount Sinai Health System. So our first couple of weeks were at Mount Sinai Hospital alone. By mid-April, we had rolled out to all of our system hospitals, and were transfusing throughout the system. We reached our 200th convalescent plasma transfusion towards the end of April, 300 in early May, and got to 400 by mid-May. And then we got lazy and took the summer off. Took us till October to get to our 500th transfusion. As you can see, we've had a little bit of a mini second wave here in the state, less so in the city. And so our transfusions have gone up in this sort of second wave, but not nearly to the extent that we saw in the first wave back in the spring. And so the EUA was announced by the FDA on August 23rd. We are currently transfusing under the transition period before the EUA. We are using up our existing plasma supply before we start switching over to purchasing EUA compliant plasma, or obtaining EUA compliant plasma through New York Blood Center, which is our blood collector. So we did an analysis of the first 39 patients that were transfused under the EIND, and that was just a sample of convenience. They were mostly middle-aged, about 2/3 male and 1/3 female, on average obese. And based on the literature for convalescent plasma for other infections, primarily influenza, SARS coronavirus, and some literature from the 30s about pneumococcal pneumonia, we thought that the most likely benefit from convalescent plasma would be as an antiviral, so that it would be most useful while the virus is actively replicating in the patient, and that once immunopathology has started to arise, and the immune response was going a little haywire, that it would be less useful in that setting. So we were focusing on patients who were early in disease. So we had a median of 7 days of symptoms prior to admission. And we were also working very hard to get to patients quickly upon admission. And as our program got underway, we were able to get the systems in place where we could get plasma to patients within a day or two of admission. And of this group, the vast majority were on some form of high flow oxygen at that point. And again, we were trying to catch people before they had gotten to the point where they needed mechanical ventilation. But these were clearly patients in acute respiratory failure who we wanted to try to prevent, ideally, from requiring mechanical ventilation. So this was all compassionate use treatment data. So in order to find a control group, we retrospectively used propensity scoring to match our 39 recipients to 156 controls. They were untreated but COVID confirmed patients who were admitted during the same time frame as our EIND recipients. We had a large number of patients to choose from, over 4,000, largely because we just didn't have the plasma supply to treat that many people at that point. So we had far more untreated people than treated people. But we were able to match all of our patients, our convalescent plasma recipients, in 1:4 ratio by propensity scoring. And as you can see in the blue here are the distribution of propensity scores for the convalescent plasma recipients. And then in red here under matched is the matched, the 156 propensity score matched controls with a very similar propensity score distribution as our recipients. And that's in comparison to the propensity scores of all 4,000 patients from which we drew the controls, were much more widely spread. So we were able to identify controls that seemed to have clinical characteristics that predicted convalescent plasma treatment, but were not treated. And we were also able to enforce exact matching on several clinical parameters, including the oxygen requirement on the day of transfusion, which for the control patients who obviously didn't get transfused, that was defined as the duration of hospitalization that their matched convalescent plasma recipient had before their transfusion. So if a recipient received a transfusion on day 3 of their admission, then the matched controls also received plasma or would have received plasma on day 3. And we use the oxygen requirement of the controls on day 3 as the starting point for day 0 for these calculations. We were able to match on intubation status as well as hydroxychloroquine and azithromycin, which were being used widely at the time. And other drugs were not being used frequently enough to enforce matching. So for the survival probability, in the 1 to 4 matched controls in blue, you can see that the convalescent plasma recipients in red did better, statistically better in terms of survival probability. And this analysis was adjusted for data that could not be ascertained just from the database alone. We had to manually collect the duration of symptoms for all of the controls, as well as other drugs that were administered, primarily therapeutic anticoagulation. We also did a Cox model for hazard ratio for in-hospital mortality, which again was adjusted for duration of symptoms prior to admission and other drugs administered during the hospitalization. And so the hazard ratios in the 1 to 4 matched cohort with replacement, which means that a given control could be matched to more than one recipient if they were the ideal match to that recipient. Adjusted for the covariates that I mentioned, duration of symptoms and other drugs received, we had a hazard ratio of 0.34 in favor of convalescent plasma recipients. Without covariate adjustment in the 1 to 4 matched analysis, remains statistically significant with a hazard ratio of 0.39. And in the 1 to 2 matched without replacement, which means that each control was matched only to one recipient. And we took the top 2 matches for each recipient, which increases the precision of the estimates at the expense of less statistical power. We saw a trend towards benefit with the hazard ratio, but the confidence interval was not statistically significant. And we also, in this Cox model, saw trends, non-significant trends towards benefit, particularly in patients who were not yet intubated, patients who were treated within 7 days of onset of symptoms, and patients who were also on therapeutic anticoagulation at the time. And there was no evidence for confounding of any of these covariates in this analysis. On a larger data set of 427 transfusions that was done by our blood bank, we had 55 reactions that were temporarily coincident with transfusion. About 3/4 of those were determined to be due to underlying disease. So a person who came in with a fever and was spiking fever throughout their admission and during their plasma treatment as well. That includes progression of respiratory disease, which could be a little bit difficult to distinguish from TRALI in certain cases. And also, some of our patients did actually have sepsis and got hypotensive, which can be a little bit confusing with TACO. So there was a lot of difficulty with the particular symptom constellation of COVID, sometimes discerning what was a transfusion reaction versus what was underlying disease. But based on this analysis, the incidence of transfusion reactions was 13% overall, and 3.1 that were thought probably due to the transfusion rather than to COVID. And of the serious reactions. About half of them were febrile non-hemolytic transfusion reaction. So not, the most severe form of transfusion reaction. In terms of the severe ones, we had 5 episodes of TACO, 1 allergic reaction, and 1 hypotensive episode. And then a little bit of unpublished data. As we moved on in our convalescent plasma treatment program, we started assessing for convalescent plasma recipients, whether they already had started mounting an antibody response prior to transfusion, with the idea that, if somebody already had started their own humoral response, giving them more antibodies may not be actually that beneficial. However, because of the assay that we were using, it took about 3 days turnaround time for us to get these results, and we couldn't act upon them clinically, but we were sending them more for research purposes to understand. And as our lab was able to implement some automation, robotic arms, things like that to speed up this process, we were able to get this information a little bit sooner. And in this analysis of 62 convalescent plasma recipients, what we saw is that patients, their pre-transfusion anti-spike IgG titer had actually a very weak correlation to symptom duration before transfusion. We had patients who, said that they were sick for 2 weeks and had no antibodies. Patients who had were sick for only a few days, already had pretty high titers. So what we were sort of empirically suspecting appears to be the case, that duration of symptoms, so a clinical only criterion, is insufficient for determining whether a potential recipient has already started to mount their own humoral response, and that really, in order to identify patients who have no antibodies, who donor antibodies might help, we really need to actually assess for the presence of antibodies prior to transfusion. And so we're working on this data a little bit more. This is a very preliminary analysis. So I think in terms of the sort of strengths of our data is that we were able to pre-screen all of our convalescent plasma donors and to know their antibody titers. Early on, we were referring patients who or donors at a 1 to 320 or higher. As the epidemic progressed and we got more and more donors, we were able to be a bit more picky and started referring only those at 1 to 960 and higher. So we were obtaining a reasonably high titer convalescent plasma supply. We have a large number of both plasma treated patients, we're up to about 550 at this point, and a lot of untreated patients to use as controls. Obviously, the caveats are that this is entirely retrospective. It's, well, a single system. It's, 6 hospitals within New York City. So the hospitals are, everything from a 200-bed community hospital to, a tertiary academic medical center. But all of this data is compassionate use data. It's all non-randomized and all retrospective analyses. So that, needs to be taken into consideration. But what we have seen so far is that there appears to be a modest clinical effectiveness of convalescent plasma when added to standard of care. It appears, again, not statistically significant, but of greater benefit when given early in disease to patients who have not gotten that sick yet. And this has been borne out in some other studies, some other retrospective analyses. We didn't have that many severe transfusion reactions. And that, the inability to tell whether a patient had started mounting their own antibody response prior to transfusion based entirely on reported duration of symptoms was unreliable. We are now screening potential convalescent plasma recipients with a rapid antibody test, which is actually looking for nucleoprotein rather than spike, but we did some correlations with our own assay and found that anyone who has a titer of 960 or above on our in-house assay has a positive on this rapid assay. And so, has a turnaround time of about 2 hours. So we are able to, now we are treating patients only if they have a negative antibody screen prior to transfusion. So we'll be looking at that data to see how that affects our outcomes. And, our analysis of our larger cohort is underway. It's very complicated and is being done by a very talented statistician who I can't possibly understand the things she understands, but she is working on it hard. And hopefully, we will be releasing some of that data as soon as we have it. And again, obviously our data, our experience, I think, is useful, but it is not a randomized control trial. And I think we do still need these data to really understand the efficacy of convalescent plasma. And so it was an enormous effort on behalf of all of these people. We took an army to do this, and I guess I will take any questions at whenever we have our panel.
Yeah, Nicole, thanks very much. There's a lot written about convalescent plasma, but the study you presented, I think, really helps clarify a lot of things that I had a lot of questions about. Certainly, when I say clarify, we're not done yet, as you pointed out toward the end of your presentation. I'm going to turn this over to Carol and James for any questions or comments that they might have before I've got some questions. Carol.
Yes, I actually had 3 questions, and you may not know the answers, but in your data, considering all comorbidities being equal, did you find that there was a certain age where you had better survival? Secondly, in terms of recruitment of donors, was there a certain time frame post-recovery where they would be more likely to be a donor if they volunteered, a week out or 2 weeks out, or et cetera? And then lastly, I wasn't sure if you had any hospitals in your systems that treated pediatric patients, and if there was any experience with them.
So I'll start backwards. We actually treated some pediatric patients here at Mount Sinai. 2 of them actually had X-linked agammaglobulinemia, so they basically can't make their own antibodies, and had been shedding virus for quite a long time and were not able to get rid of it. We gave convalescent plasma to them, and they tolerated it well and actually cleared. One of them appears to have gotten reinfected at some point, so we treated him again. We also treated adults with X-linked agammaglobulinemia, so that's been published as a case report. And your second question, remind me.
In terms of people volunteering for
Yeah. So definitely it looked like at least 3 to 4 weeks for optimal seroconversion. And our cohort, our donor cohort, has been described in a couple of publications by Anya Weinberg. So I'll refer you to those, but basically, we did definitely see increases over time. So when we tested somebody and their titer was too low, we would have them come back a week later, 10 days later, retest them. And more often than not, their titer was higher on the retesting. There were some people who didn't go over a certain threshold. So the people who, went to 320 and stayed at 320, people who went to 960 stayed at 960. But the vast majority of people did seroconvert. Many of them had high titers on our in-house assay, and most of them eventually got into a range where they were considered a donor at 320 or above. And the first question was the age. So our transfusion, we transfused from a 10-year-old kid up to, I think our oldest was 95. So, we have a very large age range. I don't think we have done age stratification yet for our data. I am sure that is something that is in the wizardry of our biostatistician is planned, but you know, empirically, I can't really say, you know, whether or not it seemed to help. I mean, one of the issues with our data is that most of our treatments were done in the spring when we kind of didn't know how to treat this disease. And, you know, we had a fairly high death rate, much higher than it is now, just because of the circumstances that we were treating patients under. So, you know, we're going to obviously look at some of our more recent data to see, now that we know how to treat this disease, what does convalescent plasma do in addition to the steroids and anticoagulation and things like that, you know, we didn't know to do at the beginning.
Thank you. Wonderful presentation.
Thanks. James?
Hey, Rick, I think you have another presenter. Is that right? I don't want to monopolize time if you still have to get to some.
No, monopolize some time. Our second presenter is unavailable. My guess is there's some demands that are going on in Texas that have made it really tough. So yeah, please go ahead.
I got it. Well, you know, maybe I'll editorialize first and see if we spur additional discussion. You know, to me, this whole experience with convalescent plasma has been really problematic, to be honest. You know, we have once again put ourselves in the position that we seem to continuously do this, where instead of designing and executing a well implemented randomized clinical trial where we would have, you know, really good data by this point, we're left still guessing. Really, it's anybody's guess whether convalescent plasma works at this point. We've transfused, at least from my understanding, over 70,000 people, you know, in some type of trial. Again, not randomized clinical controlled trial, but an uncontrolled trial. And you know, you can dice and slice and manipulate the data any way you like, but you know, the only early indication of clinical efficacy that seemed to be important was in comparing low versus high neutralizing antibody titers. And now even those data look like they are less impressive the more samples that are analyzed. And I know that you know, there's a feeling of, well, what harm can it do? You know, it might give benefit and it's not harmful. You know, maybe we have, I think, pretty good understanding of you know, convalescent plasma and IVIG administration. We understand the side effect profiles, and compared to a lot of drugs, it is relatively well tolerated. Nicole, I think, gave great data about the adverse events they've seen at their hospital, which is, you know, nothing more than you would expect. But I think you also have to understand what the opportunity cost is. You know, how much time and effort did we as a country spend to get to this point? It has to be well over $100 million to support a 70,000 infusion trial, to get convalescent plasma to run a trial like that. And who knows how many tens of thousands of person hours were spent to do this, to get us to the point where we still have no clue whether convalescent plasma works or not. You know, what a tragedy, 8 months into a public health emergency like this, that we still don't have the answer. And that's on us, right? We as a community should know better, and we should only focus on analyzing, you know, products like this in a randomized controlled trial. That's how we got the data on remdesivir. That's how we're getting data on monoclonal antibody products. It's how we finally were able to shoot down the idea of hydroxychloroquine being a miracle drug. If we only base our decisions on intuition, guessing, and crappy science, this is where we end up. We have to do better.
So James,
I agree.
yeah, yeah, Nicole, I kind of figured you would actually agree with that. Now, I will, I gotta tell you, your people that put all that energy into that in the middle of like the worst ever, that's pretty good on you. And as James pointed out, we're still with a lot of unanswered questions. and I guess a question to all of you, the panelists and Nicole, are the randomized control studies being done right now, or everybody's throwing up their hands and it's not being done right now? And where are they happening?
So, I mean, as an example here in New York, by the time we got a randomized controlled trial up and running, the epidemic would have been over. And, you know, there
Oh, yeah.
are RCTs in New York City, which, you know, they're still open. And by the time they started enrolling, there were very few patients to enroll. And we've seen that in a number of RCTs that have been published before, well, not a number, a couple of RCTs that were published before the target enrollment was hit. So I think I agree 100% that we, you know, as a community would have been better off doing an RCT. I think in the nature of this kind of disease, where it sort of hits hard and leaves if you have a good public health response to it, you're not going to be able to do this by yourself. I think it would have had to have been a sort of nationwide multi-center trial where everyone was using the same protocol, the same placebo, the same inclusion exclusion criteria. You know, I think a lot of the RCTs that are being done, I'm not sure how meta-analyzable they'll be, because there's a lot of differences in the protocols for these studies. And I think what would have been ideal is if we had had, you know, a national somebody, a federal agency, somebody to design a trial and to say, this is the protocol, this is how we're doing it. And basically, you know, if you're in the middle of your epidemic, you open it up, and as your patients go away, somebody else is opening it up somewhere else. And I think, you know, that's part of the reason we didn't go down that route, is because by the time we got it set up, it would have been over. And I think we also, here in the city, it was so bad. And I think one thing that we were able to offer was hope. And I agree that it's better to offer good science rather than hope. But in a circumstance where patients are terrified, families are terrified, clinicians are terrified, and nobody knows what they're doing, being able to offer something that you could at least tell the patient, I don't know if this is going to help, but it's probably not going to hurt. You know, I can't minimize that aspect of what our program gave to our patients and my colleagues. I agree that's not a great scientific reason, but I think, you know, the depths of the horribleness of the epidemic here in New York City, I think, you know, we all needed some bright spot on the horizon.
Yeah. And I understand that that's a natural urge to try and do something. I think that I mean, obviously we need to set up the capability to have a national network that can do these types of trials quickly in an emergency. But I think we really do have to set a precedent and a standard pushing back against that natural urge, because while I agree in this particular instance, I don't think convalescent plasma is associated with significant harm. I'm just not sure it does much good, and it certainly doesn't appear to do good on the level that would justify the investment that we've made in it so far at the expense of doing other things. But you know, that is not always the case, right? Hydroxychloroquine had the same rationale behind it. Well, it probably doesn't hurt, and it may help. Well, actually, it probably does hurt, right? I mean, I think the data are pretty convincing now, and I think many people would have told you that's what you're going to find, is that it's associated with QT prolongation and cardiac adverse events in people who are getting it, particularly when you combine it with other QT prolonging drugs. And I think the same thing happened with Ebola in West Africa. And again, this is, you know, personal to me, because we killed people in West Africa because, well, we have to give them something. You so let's try whatever we have. And that is wrong, right? We harmed people by doing that. And so we're going to continue potentially harming people until we can come to a consensus that experimental therapies need to be tested scientifically, not just thrown about willy-nilly in the middle of an emergency because, well, at least we're doing something. I think that's the wrong philosophy to take. And I think it will continue to keep us in this situation if that's how, the community communicates it. We need to be united in understanding that science has to drive these decisions. And science is possible. Good science is possible in the middle of an emergency if we make the investment to do it.
Mhm. No, I completely agree with you. I completely agree with you 100%. Yeah, it just would have been hard for us to do it as a single site. And, we need to figure out a better way.
And I agree, but I think the science has to become more, the process of the science has to become more nimble. We can stand here in November and say that, but when I look at your graph and I remember what it was like, I'm an emergency physician, what it was like from February to March. I mean, you're talking about a 6-week time frame of an illness that none of us had ever seen. And I think we can say that about a lot of the things that we were doing in discovering. You wish you could set up a trial in 6 weeks. You can say the same thing about patient proning. You can say the same thing about what kind of PPE we be wearing that's effective. Even just IV hydration. Not drowning these people in IV fluids and tolerant of hypoxia. So, I think it's an opportunity for us to make the process of being able to initiate, launch, and do quality research in a more nimble fashion. Think about it, just getting a study through an IRB, sometimes that takes more than 6 weeks, depending on where you are. And like you said, you're a single hospital system. This wasn't not a national network. But I truly appreciate the work that you've done and the data that we've gotten. It'll contribute to the data that we see in the future.
Yeah, this has been a really good, healthy discussion. And I think there were a lot of, well, first of all, I want to acknowledge there are a lot of questions in the chat. There are a lot of comments in the chat that are all valued. Because I don't have time to bring them in doesn't mean we devalue them. Understand there's just like so much time during rounds, and this discussion around the challenges of being thrown into the worst pandemic in our lifetime, not to mention in a century, presented huge challenges. And I think James, I hope you write the 3-volume set on how to fix this thing on the RCTs, because in all seriousness, that topic, it's not like people haven't thought about that before. And yet I think in the face of so many deaths so fast, the Hail Marys that went on, it's hard to, at least from my perspective, it's really hard to criticize unless you actually take a look at, okay, the consequences of those Hail Marys without science to help guide what could have been maybe potentially better outcomes sooner, which is a fine balance that is just tough. So yeah, at any rate, hey, Nicole, thank you very much. I mean, it's like you shed a lot of light on this convalescent plasma. And I as you spoke, and James, I think you'll especially appreciate this, that I recall talking to Dr. Ribner, who was treating Ebola patients at Emory, the first Ebola patients in Emory, and then talked about convalescent plasma use in Africa. And I said, well, so is it are we saving lives with this? It's like, well, flip a coin. I have no clue at all. and trying to get to a better place with that. Yeah. So anyway, let's go to the polling. And these, if you could just hang on, and I know some of you have the 1 o'clock meetings, I got that. If you could just hang on, And like, let's get through these polling questions. There are some that are really specific to convalescent plasma in terms of what's going on out there right now. It's just awareness. It's not like promoting or saying it's horrible. It's just like awareness of what's going on out there. And then there's a couple very, very important ones around PPE that we all kind of need to know where things are. So anyway, let's do the polls if you could, Amy, Amanda. And if I like zoom out because of my stability of my internet, Amy and Amanda, you can take over for this, but I think I'm good, at least for this second. In my region, the supply of convalescent plasma immediately available for administration, and they're pretty straightforward answers. All right, can we see the results from that? Okay, so 3% exceeds demand, 13% meets demand, 4% less than demand, 11% not available. All right, next. James, this is the randomized control trial right here. So far from it. Yeah.
And Rick, while people are voting, there was some comments in the comment section about how this, really had to be coordinated at a national level. And I totally agree, right? This is not, I am not pointing this out as a failure on the part of institutions that did this, that far from it. I think we heard from Dr. Bouvier the desperation that was present, that the overwhelming number of patients they were facing. But this was a failure, at a national level, that we did not assemble the required resources and direction necessary to make this happen. And that's where we need to make sure that it doesn't happen again.
Amy and Amanda, I can't see for whatever reason the how far along we are in the responses. So I'll let you make the call about when to go ahead and stop and show the results. Very, very, very few responded to this one. So I would even throw it out as a non-randomized control trial. Yeah. All right, next. This is about recruiting convalescent plasma donors. As people were responding to that, Nicole, one question I've got, it's like, and this is my limited, ED docs know a little about some things, but not much about anything kind of thing. So if I called the lab, and I said, can you do a convalescent, or can you do a antibody titer? Because I've got to figure out whether or not administer convalescent plasma. How many labs would be able to do that? I mean, that one, it's like, I have a feeling that my lab director would say what? I really took to heart your point about the timing issue. The symptoms alone are probably insufficient. Even let's say there was the RCT and it said yes, like 50% more people survive because of convalescent. That whole the symptoms alone aren't probably good enough to get the timing thing. How many labs could do that? And is that its own initiative? You would have to ramp that up. And I don't know.
So I think now there are commercial assays that are available for serological testing. At the time we were embarking on this, it was all homebrew assays through different institutions, either internally or using Florian Kramer's protocol, established, their own sort of LDT in their own lab, you know, certainly is getting more widespread. A lot of blood collectors will need to titer plasma in order to make it EUA compliant. And that's, you know, using a specific commercial assay. So for instance, New York Blood Center, which we get our blood products from, has already implemented the EUA compliant assay, and will start, you know, knowing exactly how much antibody is in each plasma unit. But again, that was something that, you know, early on, we just didn't have the capacity. A lot of people didn't have the capacity to do this.
Yeah, understand. Thank you.
You know, now there are also rapid assays too. So there's, you know, several manufacturers that have, you know, quick turnaround time, yes, no type assays.
Yeah, and as you know, we talked about, okay, like the donor program and the whole assay for the antibody and so forth. This is not like, okay, there's a drug, a magic drug that comes from a manufacturer with a vial and here's the administration instruction. This is, as James pointed out, this is a massive effort to be able to do this. So the results you see here, 13% indicated yes, they were doing recruiting convalescent plasma donors. 12% said no, 28% said don't know, and 48 without any role for the organization. Thank you. If we do the next one, I think the next few are very specific to PPE. And this is for ICU care providers and leaders, and there might be like 3 of you on at this point, and that's okay. But if you could vote, that would at least help get a feeling for where we are with the PPE. And once we see those results, if we could just, I'll let you know where the trends have been over the past probably 4 to 6 weeks in the rounds with these, because we've had these as repeated questions for the 3 series of rounds: EMS, emergency department, and critical care. We got about 20% of you voted, and we don't identify individuals for this. So like, let's see if we can get this up a little bit beyond 23%, or 20%, that'd be great. And as I've said in many of the rounds, when you see these results, we also share these here at HHS. So a sense of what's happening in real time on the ground. Hey, James, you know, I was thinking about the RCT discussion that you prompted, and yeah, I can't tell you how many journal clubs I've been in where the study with the RCT came out, and I've said, along with many others, one RCT does not alone dictate how to take care of patients the best. And so, you know, there's the one that comes out, and then there's the next one. Speaking of like remdesivir as an example, it's an evolution, and our science, it's really tough to keep up with knowing how to take care of patients the best, given our science is absolutely going to lag behind where the virus's capabilities are and capacity is. Let's stop there. N95 masks are the most common response, and then gowns. Well, actually, not short of any PPE, 19%. So you know what the trend has been in this? Early on, N95s were for sure the big one, and then over time, gowns became the most frequent response. And then we're back to N95s, and it's only like 3 percentage points, and only a few people, but still, N95s for EMS and emergency department have by far been the winner in terms of the most short. Next. This really drills down in the shortage issue. Is your organization experiencing a shortage of PPE greater than 3 days of supply, and shipment delays for your next order causing you to deploy contingency PPE preservation strategies? And this is for ICU providers and leaders. And then there's one last question after this. And we'll close out. All right, let's stop there. 25% voted. 20% said yes, 28% said no. And there's only a few who are actually ICU providers or leaders. That's fine. All right. Thank you. Next. And this is the last one coming up here. For ICU providers and leaders, our organization uses the following respirator decontamination method for reuse. And the first option is we don't decontaminate. So what I'm seeing so far is pretty interesting compared to yesterday's. We did the same question for EMS providers, and then on Thursday, we'll do it for emergency department providers as well. All right, let's stop there. And I'll just let you take a look at it right along with me. It was interesting. There were quite a few EMS providers yesterday that were using ultraviolet germicidal irradiation, then paper bag method as well. And a lot that didn't decontaminate. Actually, I think it was 48% didn't decontaminate in EMS. All right. Let's stop there. Thanks very much all for participating. That it actually is really helpful to know what's going on in the country. So I'm going to turn this back over to Carol, then James, and then Nicole for any final comments. Carol?
I just thank you for a great presentation. I hope things settle down in New York, and it's just great to hear new ideas. I know it wasn't done perfectly, but this is a once in a lifetime time in a pandemic, and I think the lessons learned will make us all grow as scientists, human beings, and physicians in the future.
Rick, I've probably said enough, but thanks for the presentation, and let's keep exchanging ideas.
Yeah, exactly. Exactly what this platform is all about. Nicole, yeah, speakers get the last word, please.
Yeah, so I think, you know, convalescent plasma, I don't think is ever intended as a destination therapy. It's a stopgap, just because, you know, nature is much faster at producing antibodies than human beings are. So I think, you know, we as an institution are, you know, moving into monoclonal antibodies and hyperimmune globulin and you know, drugs testing and things like that. So, you know, as much as we've used convalescent plasma, you know, it was never intended to be, you know, the final thing. And so, you know, we'll do our best to get what data we can from what we've done. And stay tuned for things that have better data behind them.
Yeah, thank you, Nicole. And I really want to thank the participants for joining us today. it's a topic that's sort of like people scratching their head around. And I think this conversation really helped to open that up far beyond scratching our heads. To our panelists, James and Carol, thanks very much for being on. Echo, thanks as always for the wonderful assistance and support. And Nicole, thank you so much for doing this. It's hard for me to even imagine what it may have been like in those very early days. And what you threw at it sounds like extraordinary effort. And so our thanks from all of us. Bye, and we'll see you on the next one.